Cool, right. Welcome back. Week two, me again, I'm afraid, so apologies for that. But this week is slightly different in that we will move on to looking at cell signaling. And so this begins the sort of initial stages of this course where we look at the biological processes that are required for you to understand before we then look at how tumours actually progress. So this is all about how cells signal to each other, which of course has to go wrong in cancerous cells for them to ignore proliferative signals and various other things like that. So from last week, hopefully you remember this slide, the key thing that we're going to focus on here, okay, so these are the hallmarks on the left-hand side. I've got a mouse this week, so hopefully those of you that watch the recording can actually see what I'm pointing at, is this particular hallmark, okay, that we came across last week. All of these are required, or at least most of them are required for cancerous cells to become tumorous and then cancerous, which are two distinct stages. And the first one here is sustained proliferative signaling. So what that means is that these cells have to undergo cell proliferation, cell growth, division, in the absence of growth factors, which is what we'll see in a minute. So that refers to this particular circuit here in green. And we're going to look at these two examples in a minute. And essentially, they all converge onto this one protein called RAS, which we'll look at in a lot more detail in a minute. Through lots of various cascades, what then happens is you have a change in gene expression, which is what we talked about last week. And that then allows various hallmarks, those on the left-hand side there, to become a reality. And in this case, we're talking about proliferation. Okay, so sustained signaling that tells the cells to divide again and again and again and again, forming a tumor. So it's important before we kick off with the detail that you are aware of some terminology that is used in the field. So there's something called a ligand or a ligand, otherwise known as an agonist if you do pharmacology. This is essentially just a technical term for the signaling molecule, so whether that be a hormone, a growth factor, that binds to a receptor, either on the cell surface membrane or indeed within the cell cytoplasm itself, or actually within the nucleus in some cases, and that activates downstream signaling, eventually resulting in some cellular outcome. This is typically the first messenger, not exclusively, but this is another word essentially for this. Okay, so if I use the term first messenger, that is the first signaling molecule that kicks off the process of intracellular signaling. A secondary messenger is something that the first messenger typically activates, and what we'll see is we activate a lot of these, okay, and in high quantities, which then allows rapid conduction essentially of a signal from outside the cell to within the cell to cause a response. And the whole purpose of this is to cause this, which is signal transduction. Okay, it's the word by which we mean a signal from outside the cell is somehow telling the cell what to do at a genetic level. So there are four methods of cell signaling that you should be aware of from, hopefully from A level, if not GCSE. So the first type is up at the top left here, so the sort of direct method, direct contact. What that means is that cells are interacting with each other by making physical contact. Okay, so they are introducing signals or causing signals within cells. A cell is doing that by binding to another cell. Okay. So classic example of this is within the immune system, is probably what you'll have come across in year 12. Where you have something like an antigen-presenting cell, so a macrophage or something, presenting antigen or part of an antigen to a T helper cell, and that then causes a response. It causes that T helper cell via intracellular signaling to then divide, differentiate into various subtypes and effectors. The second one is something called paracrine, which is where you, instead of having a direct contact, you have a cell that secretes some signaling molecule that travels in the extracellular matrix, which is what I showed you last week, that sort of collagen matrix, and binds to receptors on cells that are located within the vicinity. Okay, so this has to be a fairly close acting system, but that's what happens. That's the process. Okay, and this is actually the main focus of today. Okay, in terms of cell-cell interactions. The endocrine system at the bottom left here is slightly different in that you have a endocrine cell as part of a gland that will secrete a hormone, and that will travel in the bloodstream to a cell, a target cell that is much further away in the body at a distant site, and then that induces a response. And the fourth type, of course, is synaptic. Okay, so used in the nervous system, where you have a neurotransmitter released from a neuron, which then again binds to specific receptors on a target cell, causing a specific response. We're not actually going to worry too much about the direct method too much, or the neurotransmitter method. The endocrine, the paracrine systems will come across, mainly paracrine in this case. Okay, so the majority of growth hormones, growth factors, signaling that's required for cells to proliferate act via a paracrine method. Okay, so they're secreted in the local environment and act on cells locally. So in terms of what tumour cells do and what they aim to do, these are your sort of four major outcomes for, use the mouse, for what happens when a cell receives signals from outside, right, from the extracellular environment, typically via paracrine signaling. So you can either have growth factors, which cause your cells to either survive and grow, right, so to divide, to actually actively enter the cell cycle and replicate and divide. You can have other signals that cause them to differentiate, others that cause them to die, and essentially it's the cocktail of those signaling factors. It's the mixture that you have and what ratios you have that determine at any one time what your cell is going to do, right? Are we going to sit here and enter G0? Are we going to divide actively? Or actually, in some cases, are we going to die, right, and undergo apoptosis? Importantly, how this links to cancer is if you're going to become tumorous and grow, hyperproliferate, right, one of those hallmarks we talked about last week, you have to do so in the absence of proliferative signals. Okay, so if you have none of these signals here, A, B, or C, that are telling your cell to divide, okay, in theory there shouldn't be a problem. So the main issue that affects cancer cells is that they acquire the ability to divide when those signals are not present. Nothing is telling them to divide, they just do it regardless. Okay, so they sort of ignore proliferative signals or lack thereof. The thing that they must ignore, of course, are anti-growth factors. So in red, so D and E here, these are factors in the environment, TGF-beta I mentioned last week as an example, that actually right, cause a signaling cascade within a cell to stop it dividing actively. So if you have those around, not only must you, if you don't have any growth factors, not only must you actually actively proliferate in the absence of those, you actually have to ignore any anti-growth factors that are around in the environment, okay? So they have to have no effect. So hopefully what you can appreciate from this is that actually even just this one hallmark takes quite a lot of intricate detail, okay? Your cells, to become a tumour, have to do numerous things, okay? So although I was talking about lots of mutations last week, and there's a whole lecture later on mutations, it's not necessarily the worst thing in the world because we have to affect very specific processes. So a typical example of a signaling cascade, how this would work within a cell, is that you have your ligand here, your ligand. Typically, if this is binding to a receptor on the cell surface, that would be a hydrophilic ligand, hydrophilic hormone or ligand. What that will do is it will bind to a receptor that has a complementary tertiary structure to it, and that will then activate, via mechanisms we'll see in a minute, downstream signaling, okay, and it's called a cascade. What that means is we activate lots of proteins within the cell, okay, that then activate lots of other things, eventually leading to an effector being activated, okay? These come in one of three flavors. They can either be a metabolic enzyme, so an enzyme that will help and activate metabolic pathways or inhibit metabolic pathways. You could be an activator or repressor of transcription, poor translation. So you can be a transcription factor, for example. We'll come across that. So turning on genes usually that are required for cell proliferation, for example. Or you can be a cytoskeletal protein, something like actin, etc., that then causes the cytoskeleton and therefore the cell shape to actually rearrange itself in some way. So this whole process is called signal transduction. You are taking a signal that exists outside of the cell, right, this particular red ligand here, and you're relaying that information into the cell to cause a specific change, a specific response. Okay, and as we said, one of the major changes is to then cause increased proliferation of your cell. So there are two types of ligand to be aware of. If you're hydrophilic, what that means is you like water. You're not very fond of lipids. Okay, so you're by definition lipophobic. What that means is these things tend to be, like on the previous slide, that red dot. They bind to receptors on the cell surface. Okay, they don't have permission, essentially. They don't have the ability to pass through the plasma membrane effectively. If you're hydrophobic, though, so typically these are hormones that are derived not exclusively, but very often from cholesterol, they can go straight through the plasma membrane or the cell surface membrane with no problem. And so the issue there is, where do you have your receptor? You can't have a receptor on the surface if the ligand is just going to go straight through into the cytoplasm. So what happens here is that you actually, for these particular signaling molecules, you have the receptors located either in the nucleus or in the cytoplasm. Okay, and binding, forming your receptor ligand complex, then causes one of those three effects that I talked about. Typically, though, they act as transcription factors. So your receptor here, in complex with its complementary ligand, then causes an increase in transcription of genes that are useful. So what we'll look at this week, we're going to focus quite heavily on something called a G protein. So this is essentially a protein whose job in life is to bind to GTP. Okay, so similar to ATP, this is the structure of GTP, except this particular group, this organic base, is guanine and not adenine. Okay, so otherwise it's the same structure as ATP. Okay, I point out here that we've got three phosphate groups that at pH 7 will of course be negatively charged. They are denoted as the alpha, beta, and gamma phosphates, working from this side to the left of the screen here. The important thing with these G proteins is that they have GTPase activity. Okay, what that means is they have the ability, on command essentially, to hydrolyze GTP into GDP. Okay, so guanosine diphosphate, not tri. What that means is they can break this phosphoranhydride bond here. Okay, so they can cleave off this particular phosphate group. There are two types, and we're going to focus mainly on the monomeric type of G protein. There are trimeric forms as well, but we're not going to worry too much about it. So, signaling in cells, how does binding of a particular signaling molecule actually cause a response? You've got two options inside cells. The first option, I'm not going to talk about the G protein yet, is actually slightly easier to understand perhaps, which is that you are off, right, whatever this protein may be, okay, it's not functional within the cell until you stick a phosphate group on it, okay? If you're using this method, you get the phosphate group from ATP. Okay, it's not a G protein, this particular example, okay? You just phosphorylate this protein using an enzyme typically called a kinase. That's its job. It will cleave off, right? Imagine this was ATP, cleaves off this final phosphate group here and sticks it onto your protein. That then activates it, okay, by causing a conformational change. In order to inactivate the protein, you simply get rid of the phosphate group. So that's done by a class of enzymes known as a phosphatase. And that will essentially get rid of the phosphate group, and then you're back to your off state. Okay, so causing phosphorylation of your protein activates it. If you're a G protein, which is the main example for today, it's actually not that much more complicated. You are off, okay, when you're bound to GDP. Okay, so your G protein is either going to be bound to GTP or GDP. When you're bound to GDP, the diphosphate form of your GTP, you're off. And all you have to do is then essentially turn that into GTP, and then you are active, okay, as a G protein. How this becomes inactive, of course, is that I told you it has GTPase activity within itself. All you have to do is activate that. That will cleave off your phosphate group from GTP, triphosphate, and it will regenerate your diphosphate form and therefore inactivate your G protein. Importantly though, unlike this where you just get rid of the phosphate and then add it back on, the G protein differs in that you can't simply take GDP and stick the third phosphate group back onto that molecule. Okay? What you actually have to do is you have to exchange physically GDP, i.e. take it off, okay, take it from the protein, and you then have to put a new GTP molecule on. Okay? So, key thing for now, you are off as a protein if you are bound to GDP, you are active if you are bound to the GTP, okay, 3-phosphate form of that molecule. Right, the one we're going to talk about is RAS. So I mentioned that at the very start, pointing it out on the diagram. This is a monomeric G protein, as I've just shown you, okay? So all I've done here is taken an image that now calls it RAS. So again, the inactive form, it is bound to GDP. Okay, all you need to do to make it active is swap GDP for GTP, and then it's now an active protein that can then do its function, whatever that may be. Talk about that in a minute. It is helped by these two proteins here, which I'll talk about a bit later on. Okay, hopefully what you appreciate is it's quite an ask for proteins to do, especially when you need to swap GDP and GTP. It's not just a case that you can hope that a phosphate group gets stuck back on. You have to physically swap them for each other. So you need other factors that are involved. Okay, it's not just a case that we have the RAS protein. You need these things, GAPs and GEFs or GEFs, which I'll talk about in a minute. The important thing for us and for our purposes, and you'll look at this when you look at melanoma later in the course, RAS is quite commonly mutated in different types of cancer. On average, about 30%. I'll show you some more stats later. Colorectal cancer is worse. But about 30% on average of cancers certainly contain mutant RAS proteins, right? These RAS proteins are not doing their job properly, okay? There are 3 particular genes in humans that encode these proteins. You've got HRAS, NRAS, and KRAS. Don't worry about it. You've just got 3 different types. So the whole point of this is as follows, okay? You can start with a single hormone or a single growth factor that binds to a receptor, as I showed you in one of the earlier slides, and that then activates a G protein, which I've just shown you. So a given hormone binding to a receptor, forming a hormone receptor complex, can activate more than one G protein, more than one different type. But let's just assume we activate this one in the middle. What that will do, once it's activated, right, once it's in its GTP form, will then activate an effector, which then causes production of many, many, many secondary messengers, okay? Molecules within the cell that can then transduce the signal even further. And the important thing is here, you only need one hormone here, in this case, to actually activate, have tenfold induction of your second messenger, right? You've only had one G protein active, which activates this particular effector, which then itself produces, 9 or 10, in this case, secondary messengers, okay? So basically, the point is you get amplification very quickly, okay, which is what's important for cells. And each one of these, I've only shown one of them here, but each one of these secondary messengers then has a job. So one of them here might activate another tenfold number of enzymes, in this case, maybe kinases. Kinases, remember, hopefully, they phosphorylate things, that's their job. So, hopefully what you get the picture of here is we only need one binding event on the cell surface to cause a cascade and quite a dramatic amplification in the molecules that are activated within a cell, okay? So lots of product can be formed from one binding event. Importantly, there are multiple of these sort of pathways, these signal transduction cascades that converge on one single effector. So in this case, what I've shown you up until now is that you have a binding event here and we activate some effector molecule. What this diagram shows nicely is that that's just not the case within cells. You can still phosphorylate this group in this particular example by using a completely different pathway that actually phosphorylates it on a completely different site. Okay, so 2 kinases here will be activated. Number 1 for this pathway phosphorylates here, number 2 here. That's important for cell signaling because if you converge on a common effector, a common target, you can fine-tune the response. Yeah? What that means is, in some cases, you might need molecule A and molecule B to both be in the environment, and you have to have both of these phosphate groups added for that effector to become activated, to then cause cell proliferation or whatever it is. The other case is that they often act antagonistically as well. So what that means is, instead of both of these activating, let's say B here, actually causes it to be inactive. Okay? So what that means is it's the ratio of the 2 that then matters. Okay, so you can have both of these within cells, and hopefully you just get the picture here. It's more complicated than I'm telling you at the moment, but actually it allows a lot of fine-tuning, right? It's not just the case that if molecule A is in the environment, we then cause this response. You might also need molecule B, molecule C, etc. And you might need more of molecule B than molecule A, for example. Right, a definition for you is something called a protein domain, which we'll talk about next. The sort of textbook definition for this is that it's an autonomously folding part of a tertiary structure of a protein. So what that means is your protein, I've picked UVRD, it's a helicase, DNA repair helicase as an example. Your protein exists like this, and what actually happens, you think of them as a nice blob for your GCSEs and your A-levels. Actually, within that tertiary structure, you have separate substructures that start to arise, and they've been color-coded here in different colors. Okay, so this is all part of the same tertiary structure, but actually, if you were to cut, right, the bit that encodes this, 1A domain here in green, if you were to cut that off, it's autonomously folding. What that means is it will still fold up into that particular shape and potentially do its job, okay, even though it's not connected to the rest of the protein that it should be. So, a nice example here is this is the N-terminus of the protein. I've represented here, C-terminus over here. So the first amino acid to go in is here, and the last one here. I've color-coded this as a to correspond to this diagram, where you can see the domains here, still part of the same polypeptide chain, right? Still just clusters of amino acids, but they have their own roles. So the important thing with a domain is they have their own function. Okay, so don't think of a protein now as having one function. It actually has many to do its job. Okay, and I'll give you a better example in a minute. In this case, it's a helicase. So if it's going to do its job at unwinding DNA, breaking hydrogen bonds between base pairs, that's quite an active process, and you need ATP, right? So one of the jobs this protein has to do is both bind to DNA and hydrolyze ATP to get the energy liberated in order to move along and unwind your duplex. Okay, so that's called a Rec-A domain if you're interested, but it's highlighted there in 1A. So the Rec-A domain, very, very common domain within proteins. Its job is to bind and hydrolyze ATP. Okay, all of these different domains have different roles, as you'll see. I've just given you some examples. I just picked 4 proteins. Some of my favorites there. Hopefully you can see they all have Rec-A domains within them. They don't have to be at the start. They can be a bit further in. But it's quite a common protein domain. A slightly more simplistic example, perhaps, is to relate this to opening a Coca-Cola bottle. Okay, if you're the protein and your job is to take a bottle of Coca-Cola and open it, you might think you've got one function. And you'd be right in that your overall function is to open the bottle of Coca-Cola. But actually, in order to do that, you have to do 2 or 3 things, right? You have to use one hand to hold the bottle in place. You have to use the other hand to bind to the cap, not the bottle. And then you have to, get some energy from somewhere, like hydrolyzing ATP, to do the twisting motion to actually unscrew the cap. Make sense? So actually, each one of these things is a domain, right? My left hand here that's holding the bottle is a domain. My right hand that comes over the top and grabs hold of the cap is a domain. Completely different functions. One has to bind to the cap, one has to bind to the bottle. And I then might have another domain that can bind to ATP, hydrolyze it, causes a conformational change, allows me to untwist the bottle. Okay. I mention this because I'm going to use those terms, domain mainly, coming up. So one of the main receptors that you need to be aware of, commonly affected in cancer, which we'll see, is something called a receptor tyrosine kinase. Posh words, but looks like this. I've simplified it here. You have one part of this polypeptide chain that forms a domain here that allows it to bind to a ligand or a ligand. Okay, so whatever molecule it is that it's going to bind to. It then has a transmembrane spanning region here. So these are amino acids that link it to within the cytoplasm, where it then has a kinase domain. All part of the same gene, right, that's transcribed, translated. Except they have discrete functions. Okay, so part of the same protein, different functions. This guy up here, its function is to bind this particular ligand. Down here, the kinase domain, its job, kinase, remember, hydrolyzes ATP to take the phosphate group and stick it onto something else to activate it. Okay? So essentially, binding here will cause this guy to activate something else inside the cell. That's important. Okay? And again, I've put here, mutations in these receptor tyrosine kinases are very common and actually linked to many cancers indeed. Little bit more detail is that they actually act as a dimer. Okay, so hopefully you're aware from your studies at A-level, actually proteins don't exist as tertiary structural elements all by themselves. That's very, very rare. Some do, but not very many. Most of them actually have to interact with a whole separate polypeptide chain tertiary structure. So in this case, these two sort of look like lollipops here, sticking through the cell membrane, are completely separate polypeptide chains. And they have to physically interact with each other to become the functional receptor. Okay? So think of it as two sort of Lego bricks that you have to stick together to make this thing work. So it's actually a very, very common thing within eukaryotic cells. So in humans, there are 60 genes that encode different receptor tyrosine kinases. And again, hopefully you can see here in light blue, we've got the ligand binding domain or site. And then as you move through the membrane, we now have this tyrosine kinase domain. Okay, so it's going to take ATP, stick a phosphate group onto something else. How this works is as follows. That receptor is currently in its inactive form. Okay, these tyrosine kinase domains are not very good at the moment at binding to ATP and sticking phosphate groups onto other molecules. What suddenly makes them very good, okay, is the binding of the ligand. So your growth factor, whatever it is, hormone, binds to binding sites on both of these receptors, and that brings them, as you can see here, into close proximity to each other. Okay? Hopefully what you appreciate is if they both have tyrosine kinase domains here that are suddenly being brought very close together, okay, and actually they do interact physically with each other, they like sticking phosphate groups onto things. So what they're going to do is stick phosphate groups from ATP onto each other. Okay, so it's called transphosphorylation. Trans meaning across, phosphorylation, adding a phosphate group. Okay, so what you suddenly get is when your ligand is present, or ligand, I keep alternating between the two for some reason, can't decide which one I prefer. You then get this transphosphorylation event. Okay, so this tyrosine kinase domain here will use ATP and stick phosphate groups onto this. And then the same here. So this tyrosine kinase domain sticks phosphate groups onto the other one. Okay. The overall outcome is ligand binds, and then you get phosphorylation of your receptor tyrosine kinase in the cytoplasm. Hopefully what you see from this diagram is on the left-hand side, this is where we haven't yet bound the ligand. Here is when we have, and we've caused phosphorylation of the tyrosine kinase domain. And if I point it out on here, you can see we've got this loop here in red, okay, called the activation loop. Don't worry about it. It's just a bit of protein. It's just amino acids for the time being. But as soon as you stick phosphate groups here onto the tyrosine kinase domains, like this sort of transphosphorylation event, it suddenly swings this loop in a completely different direction over here. Okay, quite a remarkable conformational change within the protein. Okay, so this loop here has gone from over here and it's suddenly swung over in this direction. Okay. It's called a tyrosine kinase, receptor tyrosine kinase, because the amino acids that are very good at being phosphorylated, tyrosine is one of them. Okay, so tyrosine amino acids are what's being phosphorylated in this particular example. So just zooming into that, these little groups here with a phosphate group, it is a tyrosine amino acid within this chain that has now got a phosphate group attached to it. Okay, so it's now called phosphotyrosine. Don't worry too much about that. Key thing is we start phosphorylating this. That's important because as soon as you phosphorylate those residues, those amino acid groups within your chain here, within your cytosolic region, that then acts as a nice binding site for lots of other proteins, okay, which themselves will then potentially get phosphorylated. Okay? So just to recap so you're happy, the key thing is we bind a ligand here, that causes phosphorylation of your intracellular domains within the receptor. That then acts as a docking site, nicely here, for various proteins that can recognize certain residues here that have phosphate groups on them. So you're now recruiting a completely different protein that might now have a different role as a result of binding something outside the cell. Okay? Everyone happy that down here we're inside the cell? Yeah? Okay. What then typically happens is this is a kinase domain. So what usually then happens is it's going to stick a phosphate group onto this protein. Right? Hopefully you get the picture now. We're going to start having a bit of a cascade going on. Right, and in terms of where the RAS protein comes in, okay, same picture here, just with some names, so don't worry too much about them for now. Your ligand here binds to the extracellular domain of your receptor. You then have this transphosphorylation event here, using the tyrosine kinase domains. Okay, tyrosine amino acids now have phosphate groups attached. This allows docking of this adapter protein here. So this protein itself isn't phosphorylated, but what it does do, or what it can do, is bind to specific phosphate groups on certain tyrosine residues here. That then recruits something called a GEF, which hopefully you remember from previous. That is required to then activate your G protein here. Okay. The GEF, or GEF, stands for a GTP protein exchange factor, okay, essentially, or a guanosine nucleotide exchange factor, but don't worry too much about that. Essentially, it's going to activate your RAS, your G protein, okay? RAS is the name for the one that we're going to talk about. So all of that, quite complicated, just to activate this guy, just to activate your G protein, okay? So all of this phosphorylation binding these other proteins is going to cause GDP to be swapped out for GTP, because that's what this GEF is going to do, and that will then activate your G protein to do its thing. So just a reminder, the guanine nucleotide exchange factor, here that's on this diagram called SOS, but don't worry about the names. Okay, I've color coded it the same color, so it's orange. We suddenly activate this guy, as a result of extracellular signaling, and that is going to take your G protein here, which is shown here, and it's going to then substitute GDP for GTP. Suddenly we're now activated to do whatever job we need. Okay. Hopefully what you appreciate is it's probably going to be cell proliferation. Okay. The alternative thing is a GTPase activating protein that comes along, activates when needed the GTPase activity of the G protein, and then that can turn it off. Okay, it's important in biology that, you know, as much as you can turn things on, you have to turn them off. Okay, otherwise they're just on all the time and it's not good. Right, all this comes together hopefully, and apologies for the words. It is the naming convention here is not particularly helpful. Essentially, we've got to this point here. Okay, where we have activated, we've swapped GDP for GTP in our G protein called RAS, and that then becomes active as a G protein. Its job is to activate something else, unhelpfully called a MAP kinase. Don't ask me why. Helpfully though, or maybe not in terms of remembering it, your MAPPK3, right, MAP protein kinase, is also called RAF. That's the other name for it. Okay, so up to you which one you remember. What that is going to do, this G protein is going to cause a conformational change in this MAP kinase. And kinases, I've said it 3 times already at least, add phosphate groups onto other things. So what this is going to do is it's going to take ATP, it's going to use the phosphate groups it gets from hydrolyzing it, and it's going to stick those phosphate groups onto another kinase to activate that, and that kinase is called MAP kinase. There's only 2 kinases for that one. Okay. What that's going to do is it's going to cause a conformational change of the MAP kinase, activating it. Remember right at the start, I said we've got G proteins or we can just phosphorylate stuff, right? That then causes activation or phosphorylation and then activation of the MAP kinase, right? The MAP kinase is called MEK sometimes, and the MAP kinase is called ERK. So up to you which one you'd rather use. The end result here is that your eventual kinase here, called ERK or MAP kinase, will then phosphorylate lots of other proteins within the cell. Some of those will change other proteins, right? They will maybe activate or inhibit other proteins. Or, in the case of our purposes today, will cause a change in gene expression. Okay, will activate genes, act as transcription factors, or will activate transcription factors. Okay, so here we've got a, you know, let's say this guy here is a transcription factor. It is currently inactive, and it's only when you have signaling down this whole horrible cascade, right, RAF-MEK-ERK pathway, that you then phosphorylate it and then activate it to turn on transcription of whatever genes it targets. Okay. How this all relates to cancer is that it is this exact pathway that then causes transcription of things called cyclins, which we'll talk about next time, I think. These things called cyclins, I guess you can remember it because it sounds like the cell cycle, activate the cell cycle, right? They cause either progression through G1, or in some case re-entry from G0, and then progression through S phase. Okay, and as you will see, G1 is the single most important bit of the cell cycle because it's only in G1 that you are responsive to these extracellular signals that cause this pathway to be active. Okay, as soon as you activate progression through G1 at a certain point, doesn't matter what signals are in the environment, your cell will do one round of division. Okay, so really important. This is essentially the signaling cascade that is causing your cell cycle to be essentially turned on. In terms of mutations then, so that's the hardcore detail out of the way, you'll be pleased to know. Where does this factor into cancer? Well, if this whole process is causing cell division, it's going to cause your cell to progress through the cell cycle, hopefully you appreciate all you have to do is mutate various points along that pathway. You've got lots of options. so long as you cause the right effect, you then cause hyperproliferation. Okay. So classically, this is what happens, right? Your growth factor, whatever that may be, your ligand, binds to and causes dimerization of your receptor tyrosine kinase that activates your kinase domain, phosphorylating loads of things, eventually RAF, MEK, ERK, etc. How you get this to happen, remember we had to say, or we said, this process must happen, right? Cell division must happen in the absence of growth factors, right? So if we get rid of the growth factor, this blue looking thing here, if you have mutations in your receptor tyrosine kinases that mean this kinase domain here is activated constitutively, i.e. in the absence of ligand, suddenly you're going to activate this whole pathway, and if I look back on here, go all the way down here, turn on gene expression for things like cyclins, which then progresses you through the cell cycle when actually there's no signal from your extracellular environment that you should be doing that. Hopefully that makes sense. Okay. So that's one way in which you can have these mutations causing increased proliferation, that first hallmark of cancer. Okay. If you have a mutation in the receptor tyrosine kinase, that means it's always active. Right? It doesn't matter whether you've got a growth factor there or not. Okay. So that's called ligand or ligand independent firing. The other way this happens is actually you don't have a mutation in the protein coding sequence itself causing it to do its thing. You actually just have overexpression. So that could be a mutation in the promoter region, for example, or an enhancer, something like that, that causes that gene, instead of being transcribed in the usual copy number, in the usual way, it's overproduced. So you don't just have one of these, you've got loads of them. Okay? And again, that can cause problems with protein aggregation and various things, and it can cause them to fire without the ligand being present. The other mutation you could have is that if your particular factor here is a protein signaling molecule, right, a growth factor that might be proteinaceous in nature, it can be overactivated itself. You could have a mutation causing overproduction of growth factors, which means you're again going to overstimulate your kinase and therefore cause more cell division. So, as I said, I mentioned RAS. Okay, RAS, don't forget, is the G protein involved in this MAP kinase pathway. I've put some stats there. So I said about 30% on average mutated in cancers. Actually, for colorectal, 95% have mutations in the RAS protein. Pancreatic, 45%, lung cancer, 35%. And the important thing is, so we can mutate the receptors, or you can mutate the G protein, right? And that causes it to be on and not able to get rid of the GTP back to GDP, right? It's just sitting there with GTP, and it can't actually do anything other than turn on proliferation. So in normal normal cells here, what this G protein is able to do, don't forget it's called RAS in this particular case, is it's able to activate its GTPase activity and exchange or cleave rather that phosphate group, terminal phosphate, off, forming GDP, turning it off. In cancers, your mutated RAS, which I've denoted there with a star, it is unable, the equilibrium here has been shifted massively, it is unable to hydrolyze the GTP, right? What that means is it's sitting there and it's constitutively on, right? It's going to be constantly activating that MAP kinase, which is going to activate the MAP kinase, which is going to activate the MAP kinase, which is going to activate transcription factors and turn on gene expression for cell cycle progression, okay? And then that potentially then leads to obviously cancer, right? You get uncontrolled cell growth. Now we start to get a tumor. So in terms of treatments, and again, this links back to melanoma. So hopefully the melanoma expert will talk a little bit more about therapies. Can we inhibit RAS? So this mutant RAS as a treatment. So again, your receptor tyrosine kinase here activates your GEF, your guanine nucleotide exchange factor, and that then causes activation of RAS, and then you have your RAF, MEK, ERK, etc. These things are mutated commonly in cancers. As I said, the G protein in blue and this particular GTPase activating protein can be mutated as well. If you can't activate this, you can't hydrolyze your GTP, you can't turn off, essentially. Could you inhibit it? It's been tried, so it's being currently looked into. So this is quite common, this is sort of fairly up-to-date research that's going on. We use these things called small interfering RNAs, so siRNAs, essentially to turn off protein synthesis is the idea. So can we target this directly? All well and good turning off RAS, right? Being able to stop it being actually translated or to do its function. The problem is, although I've explained it like this, and if you're looking at the next slide already, you'll see, the problem is that RAS doesn't just activate the MAP kinase pathway, or in this case, MEK-ERK, right? It activates at least 2 other pathways, if not more. Okay, so it has very broad-ranging effects. It doesn't just activate this one particular pathway. Okay, this is the main pathway involved in cell proliferation, hence why I talked about it. But it does affect other things as well. So the problem is, if you give a therapy to someone, you're injecting them with siRNAs to try and stop production of RAS, it's going to affect all these other pathways, not just in cancerous cells, but in your normal cells as well. Okay, so actually, as I put here, it's actually not, if you read the literature on this, it's not actually been that successful in vivo, okay, in animal studies. You could target these factors that help the G protein do its thing, right? So either the exchange factor that's going to swap your GTP for GDP, or the GTPase activating protein that's going to help it essentially turn off. But the problem with that is that most mutations in RAS that we see in cancers make them insensitive to this GTPase activating protein, right? As I said, they sit there holding on to GTP, not able to do anything. What that means is they're permanently bound. So actually, if you target these Jeffs, right, that want to take away your GDP and put GTP in there, all well and good, except the problem is they're binding so tightly to GTP that's not really very efficient. So actually, the other option here is to use something called an antagonist, which is essentially a molecule that binds to a receptor or a protein in place of the normal ligand, in place of the normal thing. It doesn't actually do anything. It doesn't activate the protein. The problem with that is that even if you have picomolar amounts of GTP, that's all you need for RAS to be active, in a reasonable amount. Okay, it binds so tightly to GTP that even if you try and outcompete binding with something that's not going to be hydrolyzed, right, a GTP analog, right, something that looks like GTP, but when RAS binds to it, when the G protein binds to it, it can't do anything with it, right? It doesn't actually cause activation. The problem with that is you have to essentially flood these cells with so much of it, because even picomolar amounts of GTP, which you will have in cells anyway, actually keep RAS pretty active. Okay, so again, it's being looked at, but not perhaps the best therapeutic strategy. Could you inhibit downstream proteins? So, RAS isn't your only option, of course. Could you inhibit something along route? So either the kinase, the kinase, or the kinase, right? Could you try and stop one of them from actually having a function, right? Stop the path, instead of cutting someone off at the legs, cut them off at the knees, right? Hopefully the same idea. So the answer is yes. All of these in boxes here are various drugs. So you can see here in blue, lots of protein kinase inhibitors affecting things like MEK, okay, the MAP kinase, or RAF. Lots of these things. Anything that ends in nib, by the way, is an antibody, but we'll talk about that. So these are all being looked at. These are all in circulation. So yes, the answer is we are looking at inhibiting these. Certainly, the issue is, of course, in cancerous cells only. That's the idea. That's the problem with this, but hence why chemotherapy, etc., isn't very good. or certainly it has wide-ranging effects. The final thing, really, is talking about something called HER2. So these are your receptor tyrosine kinases, which we've covered in broad terms today. As I said, there are 60 genes that encode these things, 2 of which are called HER1 and HER2. Specifically, we're going to look sort of at HER2 here. One of the main mutations in breast cancer is overexpression of HER2. So if you hear people talk about HER2 positive or HER positive breast cancer, it tends to mean that the mutation is actually an overexpression. We've got too much of this particular receptor. And then obviously it's going to become activated, RAF, MEK, ERK, as I've told you. So we're using monoclonal antibodies. This is where these things come in, right? If you want to target the receptor itself, which is a protein, design some antibodies against the protein. The antibodies bind, and therefore your ligand or ligand can't bind. That's the idea. Okay. So you might have called this. You might have heard this is called Herceptin, maybe if you've looked into breast cancer at all. The sort of actual scientific name is. Do I try and pronounce it? Transutamab. So again, if it's an AB, it's an antibody. AB for antibody. You've also got tyrosine kinase inhibitors that are being used for this. So not only are we going to try and use antibodies to bind to HER2, we can also use these particular inhibitors of receptor tyrosine kinases as well. Again, lots of drugs are being developed. The reason is they're very complicated signaling pathways. We have to be very clear about what we're trying to target. Off-target effects are obviously potentially a problem, but where it's at really is antibody therapy or immunotherapy as well, which we'll talk about later. You don't need to know this. I point this out, just for ease. We've talked about a tiny little bit today of this, right? So you've got some kind of receptor tyrosine kinase here, activates this. What's the word? Adapter molecule, activates your RAS, right? Your G protein here, signals through MEF and ERK. Here we go, MEK, ERK, and causes transcription of these particular things. But that's not the only thing, and there's lots of crosstalk. Okay, so you don't have to worry about this at all, really. I'm just pointing out that it's far more complicated than even I've told you today. Lots of interacting factors, hence why developing drugs against these things can sometimes be a problem. Right, so I hope I told you last week. Last week was the easy week. Hopefully, again, the sessions are recorded, so I will send the links out to that. Hopefully, what you get from this particular session is that signaling is a complex process. It involves lots of cascades, right, where one protein activates another, which activates another, which activates another. That's usually quite good. It's quite useful because it allows fine tuning, right? Remember that crosstalk thing. It's quite a useful thing for cells to do. G proteins are one major system that's used to relay information, to transduce extracellular information to the cytoplasm and the nucleus to turn on various genes. The genes we looked at today were cyclins, for example. They will cause the cell cycle to progress. Autophosphorylation or transphosphorylation. The whole point of that is to provide a docking site for lots of other proteins within the cell. The examples that you probably should be aware of in terms of the course in future weeks. Epidermal growth factor receptor is the ligand that tends to activate the MAP kinase pathway quite often. And of course, that then causes cell proliferation. Lots of current research is looking to target various aspects along route. And of course, antibody therapy in HER2 breast cancer as well. Okay, so it's not just a case that we target intracellular molecules, we sometimes try and target the actual receptors on the surface as well. Before you go, there is no session next week. It is in about 2 or 3 weeks' time. So I think the 4th of November by my account. Again, back in here. Otherwise, thank you very much. Any questions? Happy to take them. Cool, thank you.
you realise that?
What? Like, in the face?
Yeah, you like, but you're doing it like crap by your
Oh, really? Can it line up?
Yeah, I was thinking, how like if Apple and they something that would like ultra wrap or something or cheaper, how would they get it? So it doesn't affect like the whole
Very tricky.
Is that like how what ways?
I mean, yeah.
Other than
So
like physically, like if you change that thing.
I just realised I'm not towards this.